Imagine you are a newborn neuron, just, trying to figure things out in a developing brain.
Right. It is an incredibly busy place to be.
Exactly. You are surrounded by this chaotic, bustling soup of, growth signals, chemical whispers, and all this structural scaffolding.
Yeah.
And amidst all this noise, you have to make one singular, critical choice.
Yeah, and it's a choice you can't really take back.
No, you can't. You have all these tiny little branches called neurites reaching out in every direction, exploring the dark. But only one of your many branches will become the all-important axon.
Right, that long, heavily insulated transmission cable.
Yeah, the cable that will carry your messages to other cells for literally the rest of your life. So how do you choose?
It is arguably the most important physical decision a neuron ever makes during its entire development. I mean, if a neuron wires itself incorrectly at this stage, the whole circuitry of the brain is compromised.
That's that high stakes, huh?
Absolutely. The downstream flow of information, it relies entirely on that single unidirectional axon acting as the central highway.
Well, today's deep dive is into a groundbreaking 2026 Nature paper by Lin and colleagues that completely, rewrites the textbook on how neurons actually make that choice.
It really does. We are looking at an incredible stack of sources detailing live cellular imaging and genetic manipulations in cortical pyramidal neurons.
Our mission today, we are going to decode the internal biological clock, or oscillator, that allows a neuron to independently make this vital decision. It completely flips what we thought we knew on its head.
Yeah, it shifts the power dynamic completely.
Okay, let's unpack this. To really appreciate this massive new discovery, we first have to understand why the old scientific model just doesn't quite add up anymore.
For decades, the dominant theory in neurobiology was this growth cone-centered view.
Right, the growth cones being the tips of the branches.
Exactly. The idea was that the tips of these little branches were essentially sniffing out external cues in the environment.
Like little bloodhounds.
Pretty much. Researchers focused heavily on extracellular matrix components or, growth factors like NT3 or TGF-beta drifting around in the brain tissue.
So the running assumption was a simple cause and effect, right? The environment just dictates the growth.
Yeah, the thought was that whichever branch sniffed out the highest concentration of these external growth factors would just automatically become the axon.
It was viewed as a totally outside-in process. The environment dangles a carrot, the branch reaches for the carrot, and boom, you have an axon.
This raises an important question, though.
Which is?
If the developing brain is absolutely rich in these growth-promoting factors, essentially bathing all the neurites in positive signals all the time, why doesn't every single neurite turn into an axon?
Oh, right, because they are all soaking in the same stuff.
Exactly. How does the cell reliably pick just one when the exposure to these signals is uniform?
The source material highlights a really glaring hole in that old theory. I mean, even when two neurites of a single neuron simultaneously contact the exact same growth-promoting substrates, only one becomes the axon.
Right, and the other is actively restrained.
I always picture it like putting a group of kids in a candy store. You tell them to grab whatever they want, but somehow, perfectly, only one kid ever ends up with candy while all the others politely put their hands in their pockets.
That is a great analogy. It completely defies logic.
The external environment cannot possibly be the only boss here. There has to be some kind of internal manager dictating who gets to grow and who has to stop, right?
Yeah, and that is exactly what these researchers observed when they started tracking unpolarized neurons using some really advanced techniques.
Unpolarized meaning they haven't picked an axon yet.
Yes, exactly. They use machine learning-assisted segmentation to track the microscopic growth profiles of these neurites over time, frame by frame.
What were they actually looking at, though? Like what kind of tissue?
They looked at unpolarized hippocampal neurons and also embryonic day 15.5 acute cortical slices.
Okay, wait. E15.5, what does that mean for the listener?
E15.5 just refers to the developmental stage in the mouse model they used, embryonic day 15.5. It's a crucial window when these cortical pyramidal neurons are actively migrating and trying to establish their polarity.
Got it. And an acute cortical slice is just a living cross-section of the brain tissue.
Exactly. It keeps the neurons in a more natural environment compared to a standard petri dish. And what they saw in these slices was amazing.
Right. Long before a neuron permanently chooses its axon, the neurites go through this highly coordinated oscillatory growth phase.
They literally take turns. It's not a mad dash to the finish line at all.
It is an intricate, microscopic dance. I mean, one neurite extends, And while it accelerates forward, the other neurites slow down or actively pull back.
Yeah, and then the extending one retracts, and another one takes a turn stepping forward.
Let's just pause and summarize where we are for the listener really quickly. We used to think the environment just yanked an axon out of a cell. But now, by watching live cells frame by frame, researchers saw the branches taking turns growing and shrinking.
Right, and the correlation analyses of these extension and retraction dynamics show this isn't just random jittering.
It's mathematically predictable, right?
Very much so. During the extension phase of one neurite, the others slow their retraction. During the retraction phase, the extending neurite decelerates.
It is a perfectly coordinated system of transient, temporary polarization. The whole cell is basically pulsing in a rhythm.
It's beautiful, really.
But wait, I'm stuck on something here. If these neurites are located on completely opposite sides of the cell, taking turns extending and retracting, there has to be a metronome keeping the beat.
That's the million-dollar question. Where is the conductor of this orchestra?
Yeah, because if the tips of the branches are just the instruments, they can't be coordinating with each other from that far away across the cell.
The researchers realized the coordination wasn't happening at the tips of the branches at all. It was happening at the very center of the cell, the soma, or the main cell body.
To figure this out, they had to look at the cell's internal skeleton, right?
Right, specifically the actin network. They introduced a genetic tracker called LifeAct into the brains of developing embryos.
LifeAct, like it acts on life.
Sort of. It basically acts as a fluorescent beacon that binds to F-actin, the structural filaments of the cell. It makes the cellular skeleton glow in the dark so they could watch the structural fluctuations in real time under a microscope.
Imagine watching a weather radar on the local news. They saw these bright, glowing patches of actin forming right in the center of the soma.
And then they noticed something truly amazing.
What was it?
In the vast majority of their observations, following a slight decrease in the brightness of these central actin patches, a literal wave of actin would shoot out from the cell body and travel down one single branch.
The timing of these physical waves is what completely breaks this mystery open, isn't it?
It is. These actin waves form at a frequency of about 2.6 waves per hour.
2.6 waves per hour. And that frequency perfectly matches the 25-minute intervals they recorded between the physical extension and retraction phases of the neurites.
Exactly. The actin wave shooting out of the soma is the actual metronome.
I want to make sure I'm picturing the physical forces here. How does a wave of protein traveling from the center of the cell actually force a branch to grow, while simultaneously telling the other branches to shrink?
It comes down to understanding the default mechanical state of the neuron. The researchers describe a state of global inhibition.
Global inhibition. So by default, the cell is highly contractile.
Yes. It is constantly under tension, constantly wanting to pull all its branches inward.
But when this actin wave travels from the soma and hits a specific neurite tip, it acts as a local activator, right?
Exactly. It temporarily relieves that global inward tension just at that one specific spot, allowing that single arm to briefly surge forward.
So the whole cell is basically a tightly coiled spring, constantly wanting to crush inward. But the center builds up this actin patch, fires a wave down arm A, and that wave temporarily blocks the shrinking force.
And so arm A stretches out, exploring, while the rest of the cell is still pulling back.
Then the wave dissipates, arm A gets yanked back by the spring, and the soma fires a new wave down arm B.
It is an incredibly elegant way to ensure that only one branch is ever exploring the environment at a single time.
And importantly, once the neuron actually commits and a true axon is chosen, this dynamic changes completely, doesn't it?
Oh, totally. In live imaging, once the axon was definitively established, it stopped receiving these waves altogether.
It just elongated persistently on its own.
Right. And the minor neurites, the branches that didn't win, continued to receive waves, but their retraction rates actually increased.
So the winning axon essentially decouples from this wave-driven oscillatory growth and takes on a life of its own.
It does.
Let's do another quick recap for the listener before we dive into the molecular engines. The whole cell wants to shrink. The center sends out a protein wave every 25 minutes that temporarily blocks the shrinking in one single arm, creating this pulsing dance.
Perfect summary. Now, to really understand this, we need to look closer at the molecular engines driving this process.
Right, because an actin wave doesn't just magically move on its own, and a cell doesn't just pull inward by magic.
There is an intense biochemical tug-of-war happening here between two highly specific protein networks.
On one side of this battle, generating the outward push, we have a protein complex called ARP23.
Right. This complex nucleates branched actin networks.
Nucleates, you mean it builds them?
Basically, yes. It promotes the rapid assembly of these actin filaments, acting as the engine. It forms these actin patches and physically drives the wave forward.
Okay, so that's the push. On the opposing side, providing that global inhibition, that inward pulling force, is actomyosin.
Yes, specifically involving myosin II. This is a contractile network. It acts as a restrictive tightening net stretched across the entire cell membrane.
I picture this exactly like a car's engine fighting against a heavy, rusted emergency brake.
I love that analogy.
The actomyosin network is the emergency brake applied to the four wheels of the car, constantly creating friction and pulling inward.
And the ARP23 complex, creating this actin wave, is a sudden, powerful rev of the engine applied to just one wheel.
It overcomes the brake for a brief moment, pushing that one wheel forward just a little bit.
The researchers actually tested that exact mechanical relationship by using a specific chemical compound.
What did they use?
A drug called CK666. It specifically inhibits the ARP23 complex.
So they essentially introduced a drug to young, unpolarized neurons that completely stall the engine.
Exactly. These were DIV1 neurons, meaning they had only been growing in vitro for 1 day.
If the engine is stalled but the emergency brake is still engaged, what happens to the cell?
The emergency brake completely takes over. Without the ARP23 local activation to push back, the developing axons and neurites completely retracted.
Wow. The global contractile force of the actomyosin net just pulled everything inward into a tight little ball.
Yeah, it just collapsed. But interestingly, when they washed the chemical blocker away, the ARP23 engine recovered, and the neurites robustly grew back.
That's wild. Did they grow back in the same way?
Yes. Among the neurons that had retracted their dominant branch, more than half actually regrew their original axon.
Wait, really? So this system retains a memory of its structural polarity even after it collapses?
It seems to, yes.
Okay, so they proved the engine is required for the push, but they went a step further to show who was really driving the car, didn't they?
They did, using optogenetics, which is just brilliant.
Using light to control cells.
Exactly. They introduced a tool called PARAC1 into the cells. This allowed them to activate the ARP23 engine using light.
By flashing a highly specific 445 nanometer laser right at the soma, the center of the cell, they could artificially trigger an actin wave on command.
What's fascinating here is the location-dependent response of that laser activation.
What do you mean?
Well, when they triggered that wave artificially at the soma, it immediately caused all the neighboring neurites to retract.
Because it turned on the whole system's dynamic.
Exactly. Then, as the artificially created wave moved away from the soma and traveled down a specific neurite, that neurite prepared to grow.
So they perfectly recreated the natural oscillation they had observed earlier just by playing the metronome themselves with a light beam.
They essentially just hijacked the cell's machinery to prove their point.
They literally took a laser, zapped the center of the cell, and forced the branches to dance to the tune of the light. That is incredible.
It's some of the most elegant experimental biology I've seen.
But, it still leaves me with a massive question.
Go for it.
If this entire process is just a continuous, endless loop of engine revs pushing out and emergency brakes pulling in, how does the neuron ever finally commit?
You're looking for the mechanism that breaks the cycle. How does one branch choose to become the single axon and establish permanent polarity?
Right. If it's just a tug of war, how does anyone actually win the war? The sources describe something called the escape mechanism.
Yes.
Yeah.
When that actin wave finally reaches the very tip of a neurite, it temporarily relaxes the local actomyosin brake.
And in that brief window of relaxation, a new structural player enters the game.
Microtubules.
Let's talk about microtubules.
Microtubules are the heavy-duty structural support beams of the cell. While the actin networks we've been discussing are highly dynamic, constantly remodeling and turning over very quickly, the microtubules provide rigid, long-term stability.
Okay, so during that brief transient extension caused by the actin wave, these rigid microtubules protrude forward into the newly created space at the tip of the branch.
Exactly.
Let me update my car analogy.
Yes.
The car engine revs, fighting against the heavy emergency brake. The car surges forward just an inch or two.
Right.
But during that brief surge, the microtubules act like wet concrete poured directly behind the tire.
Wow, that perfectly captures it.
The concrete sets quickly. Once the microtubules stabilize enough inside a growing branch during one of these transient pushes, that neurite becomes physically resistant to the global actomyosin retraction.
The brake is still pulling, but the structural integrity of the branch is now too strong to be pulled back.
It quite literally escapes the oscillation.
Yes, the emergency brake can't fight the concrete.
And once it escapes, it doesn't even need the engine-res the actin waves to grow anymore, does it?
Nope, it just takes off persistently on its own, building more and more microtubule highway.
While that winning axon escapes and grows, the fate of the remaining neurites is permanently sealed.
It is. The actomyosin activity actually reinforces its suppression on those remaining branches, dooming them to become dendrites.
They are permanently held back by the global inhibition, never quite able to get enough microtubule support to break free.
And the researchers proved this default restriction beautifully. They treated the cells with a different drug this time, a myosin II inhibitor called blebbistatin.
So they chemically cut the emergency brake.
Exactly. And without that baseline tension holding the cell together, the physical behavior of the neuron completely transformed.
The cells grew longer, multiple neurites all at once.
Yeah, without that default state of holding growth back, all the branches just started growing wildly without needing the actin wave to push them.
It proves that the neuron's natural state isn't to reach out and grow. Its natural state is to hold itself back.
It tightly controls its own structure, only releasing one carefully chosen branch that manages to build enough concrete support to escape.
This internal soma-driven mechanism is what ensures the emergence of a single axon, completely independent of those environmental cues we talked about at the beginning.
It underpins the entire unidirectional information flow in neuronal circuits. I mean, if the neuron just relied on external growth factors, as the old textbook model suggested,
It would sprout extra axons everywhere in response to the environment.
exactly, creating total chaos and short circuits in the brain's architecture.
So what does this all mean for you, the listener?
It means the neuron is not a passive listener, waiting to be told what to do by the surrounding tissue.
It is a highly active, fiercely coordinated machine. It relies on an internal cytoskeletal oscillator, a brilliant, relentless tug-of-war between waves of actin pushing out and a contractile net pulling in to guarantee that only a single, perfect axon emerges to wire your brain.
And if we connect this to the bigger picture, this discovery is profoundly important for modern medicine.
How so?
Beyond just solving a foundational mystery of how the brain wires itself during early embryonic development, understanding this precise balance between the actin and microtubule systems is crucial for neural repair.
Ah, I see. If we know exactly how the cellular brake and the engine work, we might be able to fix the car when it breaks down.
Precisely. Modulating this exact cytoskeletal system could reveal entirely new therapeutic strategies for regenerating axons.
Like in spinal cord injuries.
Yes. When a patient suffers a severe injury to the adult central nervous system, like a spinal cord injury, the axons are severed. And they struggle to regrow because the cellular environment in an adult becomes incredibly inhibitory.
The emergency brake is pulled all the way up permanently.
It is. By understanding how to artificially relax that actomyosin brake using targeted therapies, or stimulate the ARP23 engine and microtubule stabilization.
We might be able to coax mature neurons into escaping their inhibition and regrowing damaged connections across the injury site.
We are talking about unlocking the cell's own internal machinery to force it to rebuild a bridge it thought was permanently closed.
That is a massive paradigm shift for regenerative medicine, which leaves us with a final thought to ponder.
Let's hear it.
If this highly coordinated, microscopic internal metronome is what flawlessly wires our brains before we are even born, what happens when this biological clock skips a beat?
That's a chilling thought.
Right. We just talked about repairing injuries in adults, but think about the very beginning of life in the womb. Could subtle genetic glitches in this precise molecular tug-of-war, like a cellular brake that pulls too hard or an actin wave that fires too slowly, be the hidden starting point for certain neurodevelopmental disorders?
It's entirely possible.
If the structural rules of the cell are rigged from the start, how does that alter the whole physical architecture of the human mind? Something to think about the next time you learn a new fact, form a new memory, and fire a signal down those perfectly chosen, perfectly timed axons.